Okay, I think we'll get started. Hello, everyone. My name is Lisa LaValle. I'm the Director of Marketing and Communications for Chemistry of Life Processes Institute. And we're really thrilled to welcome you to our first Faculty Chalk Talk of the year. And our guest is Xiaoyu Zhang. He is one of our newest faculty members, and we're really excited to have him today. Xiaoyu's topic of his talk today is expanding the landscape of E3 ligases for targeted protein degradation. Xiaoyu joined Northwestern last February, and previously he was a Damon Runyon Fellow in Ben Kravitz's lab at Scripps Research. He earned his PhD in biochemistry and molecular biology from Cornell University in 2017. So before we get started, I just wanted to remind you of a few things, that microphones will be muted throughout the presentation, and there will be 15 minutes for questions and answers after the presentation. And at that time, feel free to turn your video and your microphone on to ask a question, or you could submit your questions using the chat feature at the bottom of the screen. This program will be recorded and posted on our CLP News YouTube channel. Anyway, so with that, it's my pleasure to welcome Professor Xiaoyu Zhang.
Okay, good afternoon, everyone. Thank you for attending this Chalk Talk. So it's my great pleasure to be here today. And as Lisa introduced, my lab is a chemical biology lab. And in general, we are interested in making small molecules to modulate the protein functions in different ways compared with conventional small molecule inhibitors. So for today's talk, I would like to start with a very general question I have been thinking about it since I was a postdoc. How can we expand the druggable proteome? And I think right now we have pretty good knowledge of human genetics. For many human diseases, we pretty much know the genetic alterations that cause those diseases. But in terms of therapeutics, there's a huge gap translating this good knowledge into effective therapies. And if you look at this pie chart, among 20,000 proteins, about a quarter of them are disease-related. And of course, we have been trying to discover small molecules that can act on those disease-causing proteins. But the challenge is that only a small proportion of those disease-causing proteins is amenable for small molecule therapies. And as a consequence, we often call them druggable proteins. But as you can see here, there are still a large fraction of disease-causing proteins that are thought to be undruggable because no small molecules can disrupt their functions. And a part of the reason is that we have been thinking about small molecules in a relatively one-dimensional and a limited manner. So we typically want small molecules to bind hydrophobic pockets on target proteins that encode some sort of biochemical activities. So very much like a keyhole and a key. We want to lock down the biochemical activity of the target protein. And this actually works very well for proteins like enzymes and receptors. The problem is that many disease-causing proteins do not have those binding sites, such as transcription factors and adapter proteins. So the big question is, what is the strategy to work around those proteins? It turns out that one source of inspiration comes from several serendipitous discoveries. So what you see here are the chemical structures of immunomodulatory drugs, or IMIDs, that have been used in clinic for years to treat multiple myeloma, but also cause this horrible teratogenic effect. And for a long time, we didn't really understand the mechanism until a few seminal studies uncovered a very unique mechanism of action for those compounds. It turns out that IMIDs bind a protein called cereblon, which is an E3 ligase that controls protein degradation. And instead of inhibiting this E3 ligase, those compounds can create new surface patches on cereblon. And then what happens is that the compound bound cereblon can interact and degrade a new substrate, such as IKZF103 or CL4. So the degradation of those proteins can explain why the same type of drug has both a therapeutic and a severe side effect. And now we call this mechanism a molecular glue type of mechanism because this molecule sits at protein-protein interaction interface and functions like glue to stick two proteins together. Importantly, if you look at those degraded proteins, they are all transcription factors, which are historically thought to be undruggable. But now we have a new strategy to target those challenging proteins. A similar strategy was actually proposed even earlier and pioneered by Craig Cruz's lab using a rational approach. So in this approach, a bifunctional molecule, also known as Proteolysis-targeting chimera or PROTAC, is made by connecting two moieties that bridge an E3 ligase and a protein of interest. So similar as molecular glues, bifunctional PROTACs can hijack E3 ligase to promote target degradation. So this is a very active field in chemical biology and drug discovery in the past few years. So here I just want to highlight one PROTAC example reported back in 2008 by Craig Cruz's lab, which is one of the earliest all small molecule PROTACs. So this compound is made by connecting a ligand binding the E3 ligase MDM2 to another ligand binding the target protein androgen receptor. So this compound has been shown to induce the rapid degradation of androgen receptor in process cancer cell lines. So no matter those compounds are molecular glues or bifunctional PROTACs, the eventual consequence is to degrade the proteins. So we call those small molecules protein degraders. And this really opens up a new direction in drug discovery. Compared with conventional protein inhibitors, protein degraders have many advantages. First, protein degraders hijack the E3 ligase to induce protein degradation. So this is an enzymatic and a catalytic process, which means you can use very low drug concentration to produce a therapeutic effect. And the second, some proteins have multiple functional domains. So it will be very challenging to fully disrupt their functions by using protein inhibitors that presumably bind only one of those domains. But this challenge can be overcome by using protein degraders because they simply remove the target protein from the cells. Another advantage is that protein degraders have the potential to target proteins that have no hydrophobic pockets. As I just showed you, IMIDs degrade transcription factors. Another type of drug called sulfonamide drugs also function as molecular glues. And they have been shown to degrade RNA splicing factors that were also previously thought to be chemically inaccessible. So I just told you that protein degraders hijack the E3 ligase to promote protein degradation. So how does this hijack the system work? Eukaryotic cells use this ubiquitin proteasome system, or UPS, to control the degradation of about 70% of proteins in cells. As you can see, this is a multi-step process involving many enzymes. But I just want to point out that there are E1, E2, and E3 enzymes that play critical roles in transferring a protein called ubiquitin onto the substrate to create a modification called polyubiquitination. So then the proteasome machinery can recognize this modification and degrade the modified protein. So protein degraders hijack this E3 ligase, which belongs to a very big protein family. So in human genome, there are over 600 E3s. And then you may think, that's great. There are so many E3s we can hijack to make protein degraders. But actually, when I started working in this field, that was back in 2018 when I was a postdoc, there were only 5 E3s that can be hijacked by small molecules to promote protein degradation. But this limited number of E3s came with some challenges. For example, cereblon is not an essential gene which means cells can delete or mutate this gene, leading to resistance to cereblon-based degraders. VHL, on the other hand, is an essential gene, but many studies found that we cannot use this single E3 to degrade other proteins. So it does have its own substrate scope. So you can see that there's a clear need to discover additional E3s to expand the application of this emerging therapeutic approach. And over the last few years, there was a huge progress in this space. And today, if you look at this list again, there are at least 16 E3s that can be chemically controlled to induce protein degradation. And I was able to make some contributions by discovering 3 new hijackable E3s. And today I want to tell you 2 stories how I discovered DCAF16 and DCAF11. So back in 2018, when I started working on this project, I wondered what would be a good strategy to discover new E3s. And if you think about this design principle, it has multiple steps. And in order to discover new E3s for TPD application, all those steps need to happen correctly. So first, we need an E3 ligase that functions in protein degradation. And the second, we need a ligand binding that E3 ligase. And then more importantly, when we make bifunctional molecules, they need to be functional degraders. So given all those essential steps, there are several strategies that have been proposed. So in one strategy, people first pick an E3 ligase, and then develop the ligands for that E3, and finally make bifunctional molecules and test if they can support protein degradation. In another strategy, people focus on small molecules. And now there are different methods we can use to identify protein target of small molecules. So there's a chance we can identify small molecules that specifically bind the E3 ligases. So we can harness this small molecule E3 interaction to further develop bifunctional PROTACs. So you can see that both approaches start with E3 ligand interaction and eventually make bifunctional molecules and test them in biological systems. But sometimes, even we have a really potent and selective ligands, when we eventually make bifunctional molecules, they just don't degrade the proteins of interest. And if that is the case, it could be a very frustrating process. And then I thought whether there could be a strategy that allows us to discover functional degraders in the first place. And then we can understand the mechanism to identify the E3s involved in this degradation process. So how do we do that? A few years ago, Ben Cravath's lab at Scripps reported a chemoproteomic platform to discover proteins with ligandable cysteines. And this study identified some fragmented electrophiles, what they call scout ligands, that engage thousands of cysteines in human proteome. And many of those cysteines are found in E3 ligases. So then I thought, can I leverage those ligand E3 interactions and couple those fragments to another ligand specifically binding a protein of interest? So I can use those fragment-based bifunctional molecules to identify functional degraders, degrading that target of interest. To that end, I chose 3 scout ligands that have very broad reactivity and couple them to a ligand specifically binding a protein called FKBP12. So the idea is to test if these fragment-based heterobifunctional compounds can covalently engage any E3s and bring them to FKBP12 to degrade it. So I generated 2 cell systems, one expressing cytosolic FKBP12, the other expressing nuclear FKBP12. So we can evaluate the cytosolic and the nuclear E3s respectively. So I found that none of the compounds reduced the cytosolic FKBP12 level. But for nuclear FKBP12, I found one compound, KB02-SLF, that can dramatically reduce its level at both time points we tested. I further confirmed this nuclear specific reduction by immunofluorescence. So here you can see that without compound treatment, this engineered nuclear FKBP12 is mainly localized in nucleus with some leaking signal in cytosol. But in the presence of this degrader, this bifunctional molecule, only a nuclear signal was dramatically reduced. So there are different ways to reduce protein expression. To further understand the mechanism, I found that a proteome inhibitor called MG132 can fully block this compound-induced FKBP12 reduction, suggesting that this reduction is caused by proteome-mediated protein degradation. So one nice thing of this TPD field is that there are many very good chemical tools we can use to further narrow down the mechanism. So I further found that a nadylation inhibitor called MLN4924 that selectively inhibits choline ring type E3 ligase can also fully block the degradation, suggesting the mechanism of action may involve a choline ring type E3 ligase. Moreover, the covalent interaction is essential because if I replace this covalent warhead with a propanamide, a non-covalent group, this compound becomes totally inactive. So all this evidence suggests that KB02-SLF may covalently engage a nuclear choline ring type E3 ligase to induce FKBP12 degradation. So next, I want to identify this E3 ligase. If you think about this mechanism, in order to induce target degradation, a bifunctional degrader needs to first induce a ternary complex between the E3 ligase and the substrate. And in this ternary complex, we know the substrate, which is FKBP12. We also have the compound. So I thought, why not I just enrich this whole complex so I can use mass spectrometer to identify what that E3 ligase is. So I used a metabolic labeling approach called a SciLike to quantitatively identify the E3 ligase that is associated with FKBP12 in a compound-dependent manner. So if you look at the waterfall plot showing on the right, the high ratio values represent proteins that are selectively enriched from the cells treated with KB02-SLF. So this experiment identified only one choline ring type E3 ligase called DCAF16. And interestingly, we also identify another protein called DDB1, which is an adapter protein that is associated with DCAF16 in an E3 complex. I do want to point out that there are many other proteins being enriched in a compound-dependent manner, as we are using a fragment-based bifunctional compound. But among all those enriched proteins, DCAF16 is the only choline ring type E3 ligase. So if you Google this protein, DCAF16 is a very poorly studied E3 ligase. So there were basically 0 papers about the functions of this E3 ligase. So the first thing I did is to confirm its nuclear localization by immunofluorescence. As you can see here, DCAF16 is exclusively localized in nucleus. And then to validate if DCAF16 is the essential E3 that supports KB02-SLF-induced FKBP12 degradation, I generated a DCAF16 knockout cells and found that in DCAF16 knockout cells, this compound no longer degraded FKBP12. Importantly, if I reintroduce DCAF16 in DCAF16 knockout cells, this can restore the degradation activity. So this data taken together suggests that DCAF16 is the essential E3 ligase that supports KB02-SLF-induced FKBP12 degradation. As KB02-SLF should covalently engage cysteine residues in DCAF16, I wonder what is the modified site that is essential for the degradation. And if you look at the DCAF16 protein sequence, it has 8 cysteine residues, including 3 consecutive cysteines from cysteine 177 to 179. And using mass spectrometry, I found that this compound modified one of these 3 consecutive cysteines. And if I mutate cysteine 177 or 179, these two mutants no longer degrade FKBP12, suggesting these two cysteines are likely the modified site for target degradation. So with these findings, I wonder if I could harness DCAF16 to degrade another disease-relevant nuclear protein. So I chose the protein BRD4, which is an epigenetic reader protein that drives the progression of multiple cancers. And I made another bifunctional compound by coupling DCAF16 fragment ligand to JQ1, which is a ligand for BRD4. So I found this compound can degrade BRD4 in DCAF16 wild-type cells, but in knockout cells, this effect was substantially blocked. So now we know that we can use covalent molecules to hijack DCAF16 to degrade nuclear proteins. And one remaining important question is, what is the fractional engagement of DCAF16 to drive target degradation? To answer this question, I use a chemoproteomic method called activity-based protein profiling, or ABPP. In this method, a chemical probe, iodosadamide dithiobiotene, that has very broad cysteine reactivity is used to capture all reactive cysteines across the proteome, including DCAF16 ligandated cysteine. And each probe modified peptide will generate a specific mass spec spectrum. And with this profile, I can further test the compounds to see if they can block a probe modified DCAF16 peptide containing the ligandated cysteine. And we can further quantify that blockage as the compound engagement. It turns out that the two bifunctional molecules produced about 10 and 40% engagement of DCAF16 peptide that contains the 3 consecutive cysteines. And consistent with this finding, in Western blot analysis, both bifunctional compounds induce a fractional band shift of DCAF16, which indicates a compound modified protein. And in both cases, you can see that only a modest fractional engagement is sufficient to drive target degradation. So to summarize this work, by using a chemoproteomic approach, I discovered DCAF16 as a novel E3 ligase that can be pharmacologically controlled to promote protein degradation. So I want to highlight 3 features of this ligase that may offer advantages in this TBD field. So first, the covalent interaction between the compound and the ligase may improve the durability of target degradation in biological systems. And second, DCAF16 promotes nuclear restricted protein degradation, which may avoid degrading unnecessary cytosolic proteins. And the third, only low fractional engagement of DCAF16 is sufficient to induce protein degradation. And I think this is an important feature because it avoids perturbing DCAF16 native functions during this degradation process, as those functions can still be performed by unmodified ligase. So I hope I convince you that by using fragment-based bifunctional molecules, we can discover functional degraders. And the chemoproteomics can further allow us to understand the mechanism by deconvoluting the essential E3s for the degradation activity. So going forward, I wonder if I could expand this bifunctional compound library and the cell models to discover more E3s with differential properties. So to this end, I and my colleagues built a focused bifunctional library that has 37 FKBP12 directed bifunctional compounds. I also established 5 cell models that express cytosolic, nuclear, or membrane FKBP12. So the goal is to screen for molecules that degrade FKBP12 in any of the cell models. And to make it in a high throughput way, I fused the luciferase to FKBP12. So I can use luciferase activity as the readout to measure FKBP12 abundance. So here's the heat map summarizing the initial screening result. The blue color represents low FKBP12 level. So you can see that many compounds promoted FKBP12 reduction. So today I want to tell you one follow-up story. So I found one compound, which is 21-SLF, that reduced both cytosolic and nuclear FKBP12 levels in process cancer cell line 22RV1. So this data shows that both proteome inhibitor and methylation inhibitor can fully block 21-SLF-induced FKBP12 degradation, suggesting the mechanism of action may involve another calling ring type E3 ligase. So then I use a similar approach as I show you in the first story to enrich this ternary complex. So we can use mass spec to identify the associated E3 ligase. So this time I identified another DCAF protein, DCAF11, that is associated with FKBP12 in the presence of the compound. And the adapter protein DDB1 was also identified as expected. To confirm this result, I generated DCAF11 knockout cells. As you can see on the right, this compound didn't degrade FKBP12 in the knockout cells. So then to measure the selectivity of this bifunctional molecule, I did a whole proteome analysis. So in these graphs, each spot is a protein in the tested cell line. So the left graph is the profile in DCAF11 wild-type cells. As you can see, 21-SLF can selectively degrade FKBP12 in DCAF11 wild-type cells. But in DCAF11 knockout cells, FKBP12 level is not affected with compound treatment. So one interesting data is that DCAF11 may not be equally expressed in different cell types. For example, HEC293 T cells express low level of DCAF11, as you can see on the left. And consistent with this, in HEC293 T cells, if I only overexpress FKBP12, 21-SLF does not degrade it. But if I overexpress both FKBP12 and DCAF11, this compound shows clear concentration-dependent target degradation. So this data further confirms that DCAF11 plays an essential role in supporting this 21-SLF-induced FKBP12 degradation. So next, to map the ligandated cysteine that is critical for the degradation, I used a mutagenesis approach. So I first mutated each individual cysteine in DCAF11. However, all these single mutants are still active, as you can see on the left in this bar graph. But I noticed that one mutant, this cysteine 460 to alanine mutation, seems to partially block the degradation. So on top of this mutant, I made double cysteine mutants. But unfortunately, all those double cysteine mutants are still partially active. So then I thought that maybe cysteine 460 is located in a pocket where there are multiple redundant cysteines. Although there's no DCAF11 structure, I was able to find a distantly related homolog, the U5 subunit of human spliceosome. And both proteins have this WDR domain. So then I aligned the sequences of these two proteins and found 3 cysteines in DCAF11 that may be in proximity. So then I mutated all 3 cysteines. And this time, as you can see, this triple cysteine mutant is totally inactive. So I just show you that mutating cysteine 460 partially blocked the degradation. Interestingly, if I mutate the other 2 cysteines, cysteine 485 and cysteine 443, this double mutant is actually equally active as wild type. So I think this data means that although these 3 cysteines are redundant, cysteine 460 is still likely the preferred ligandated site. And for the future drug discovery program, targeting this cysteine might be sufficient to promote target degradation. And finally, to demonstrate that we can harness DCAF11 to degrade other disease-relevant proteins, I chose the protein androgen receptor that plays critical roles in process cancer progression. and made a PROTAC by connecting DCAF11 ligand to an AR ligand. As you can see in this Western blot, this compound induced AR degradation in DCAF11 wild type, but not knockout cells. So to summarize the second story, by screening an expanded bifunctional compound library in multiple cell lines, we identified one compound that degraded FKBP12 in a processed cancer cell line. And using chemoproteomics, identified DCAF11 as the essential E3 ligase for the degradation activity. And different from DCAF16, DCAF11 is able to support ligand-induced degradation of both cytosolic and nuclear proteins. So one interesting data I want to point out is that from the initial screen, this compound doesn't degrade FKBP12 in the other cancer cell lines. And based on CCLE protein expression profile, DCAF11 has higher protein expression in 22RV1 than the other 3 cancer cell lines. And this is quite interesting and promising to us because the differential expression of DCAF11 may allow us to develop cell type specific degraders. So right now we are looking into more cell lines to get a broader view of DCAF11 expression versus its capability to support target degradation. So finally, I want to thank all my lab members, my postdoc advisor and colleagues, my collaborators at Northwestern, and as well as funding resource. And thank you for your attention. I will be happy to take any questions you may have.
Thanks, Xiaoyu. Great talk. I've encouraged people, if they want to write a question, to go ahead and put it in the chat, or to just feel free to unmute themselves and ask and show their video and ask their question directly.
So yeah, maybe I can read the questions from the chat. I saw a question, can you create a trivalent linkers that either binds 2 sites on the E3 ligase or 2 sites on the target protein to enhance degradation? So this is a very interesting idea. I think there are a few papers actually reporting this idea. I think it's possible if we create a trivalent molecules, we can recruit multiple E3 ligases to degrade a single protein, or we can, leverage one E3 to degrade multiple proteins. So it's going to be a very interesting idea to further explore. I saw another question from the chat, how to avoid over-degradation of the targeted protein of interest. So I think typically when we design protein degraders, we want to remove those disease-causing proteins. So I would say we pretty much want to degrade all the proteins, right? So we don't need to worry about over-degrading those disease-causing proteins. But in certain cases, we probably don't want to degrade all the proteins because for certain proteins, they may have certain functions. So if we over-degrade those proteins, it may cause problems. So I think this also underscore the need to discover new E3s, because for some E3 ligases, we may tune their activities to not degrade all the proteins of interest. But right now, I think in this field, it's like if we have a functional degraders, they typically work very well, so they can remove all the proteins. But I think in the future, if we can find other E3s that are more tunable, we may be able to achieve, just degradation of proteins with certain percentage.
Any more? Going once, going twice? Okay. With that, I think we'll close, and just really like to thank Professor Zhang for joining us. And if you have any other questions, feel free to shoot us an email. Please follow us on social media so you'll know when our next talk is. And thanks again for joining us. We'll see you next time. Bye.